Hello, my name is Sam Yencho, and today I am going to be presenting on fluorosensors and their applications. So they're actually really interesting, but I will start by presenting a problem. So the problem starts in the fact that oil spills are a major issue plaguing many ecosystems in many parts of the world. Uh, there's approximately 3 or 4 per year, according to Wikipedia, and I got the list down here in the lower right corner. And it causes a lot of issues. For example, if if it spilled into a bay somewhere and it gets into the wings of a bird, it can cause that bird to be unable to fly, which causes its own plethora of issues. It messes with the mating and offspring of turtles, and in some animals who eat it, it's not only toxic to them, but to any animal who eats it in turn, including humans. It can, sometimes seafood is toxic, yeah, because of this stuff. So there are a lot of problems in a lot of issues with fixing oil spills, cleaning up oil spills. One of which is detection of them. They, if there's small amounts and they are still dangerous, it can be very hard to detect. And also they can be hard to detect in general. So solution, laser fluorosensors. Laser fluorosensors are useful in that they're very precise. They can detect even 10 to the negative seventh, 100 nanometers of oil film. And that's really small. They can also determine the film thickness and even the type of oil based on the wavelength of the light detected and such. And you can actually see what one of the devices looks like down here in the lower right corner. And this is what the oil spills can look like. So let's move on to the shortcomings of alternative detection methods. So one of them is called chromatography. Chromatography, which is this, by the way, is when you take a sample of the substance and you use a solvent to separate out its parts, such as the oil and the water. Now, this is all well and good, but it's not nearly fast enough, efficient enough, or precise enough. I mean, you can't really fly a plane over a thing and just dip a thing down, dip a film down, pull it back up, do the solvent, and do the whole experiment. And it's not precise enough. Now, other detector methods, such as microwaves and radar, rely on characteristics such as reflection of light of various wavelengths and scattering of microwaves, which are not terrible, but they're not nearly as precise, especially because everything reflects light. So it's not a unique property of oil. So let's move on to the property we can use, fluorescence. Fluorescence is a property of certain chemicals where they can absorb electromagnetism, usually in the form of either photons or a current. And then after some time, they can spit it back out in the form of other photons. It's really just transitioning from energy states with an electron at the same spin before and after transition. We've seen this with lasing and hydrogen bands and stuff, but it's here and it's a property of some chemicals. Now, oil is one of the chemicals that can emit fluorescent light, and it has a special property about it, which I'll get to soon. Now, cool, fun thing about fluorescent light is that it tends to be emitted in the form of radiant, vibrant colors, which is cool. How do fluorosensors? Well, what they do is, what we're going to take this for example, is they shoot a laser down at whatever the target is. Here it's this phytoplankton. I guess it can be used for that, but right now we're talking about oil. So then the thing emits fluorescent light, which then is directed in there using lasers or a telescopic lens, and is detected by a photomultiplier or something of the sort. So you generally it's looks more like this. I mean, depending on how high up the field of view is different. It does have to be a plane. So this generally isn't really accurate. At least in everything I saw, it has to be a plane. So this isn't really as accurate. And again, it usually is supposed to be oil. So let's move on to spectral signature. This is a unique, oil is something with a unique spectral signature, and that is what makes it so visible to a fluorosensor. Because a spectral signature is the unique way a chemical reflects or emits light. So what else is emitting light? Things that fluoresce. Because oil is emitting the light, it has a unique way of emitting fluorescent light because of its unique spectral signature. And different types of light has slightly different different types of oil have different spectral signatures, which makes them separate, which makes them have different types of wavelengths which get measured differently. So that's a useful property of oil. Now here's the thing: the system outputs, the actual devices will just detect this, a collection of light, which some of it needs to be transferred out, filtered out because it's part of the water. Some of it needs to be filtered out because it's just errant light. And this is all stuff that the pilots of the planes or whoever's cleaning, they don't need to see that. And they just need to see the results of where the oil is. So there's actually some cool advanced algorithms which plot this data geographically, usually with different colors to detect different types of, to say it's different types of oil, that's the like. And that's useful because that gives the information quickly to the humans who need it without having to calculate anything. So now we have active and passive sensors. This is actually important because fluorosensors are active sensors and in all the stuff we've seen so far. So first of all, what are they? Passive sensors are sensors that rely on external sources of light to work. For example, the sun. Passive sensors are limited by the fact that they can generally only be used during the day, because the biggest natural source of light on Earth, besides some exceptions, okay, is the sun. So there are other sources of light, like say thermal signatures, but and they can be used, but they're a lot less. They're a lot less useful because they're a lot less intense, a lot less precise and stuff. Here's the thing: active sensors, they supply their own light. They have usually lasers that can, like we saw earlier with the fluorosensor on the boat, it shoots out its own laser, which then makes the stuff emit, makes the oil emit its own wavelengths of light. So the laser it actually can be used during the day and can be used at wavelengths that the sun can't. It does have its own issues, but I will get there now, because I decided to combine these two because they're so interlinked, the future of fluorosensors and some cons about it. Now, the major issue with current fluorosensors is that they're very heavy. The power source is actually about 450 pounds on its own. Now, hopefully in the future, because there's advancements every day as it is, fluorosensors would be feasible in a biplane with a high power, high repetition rate diode pumped solid state laser. Try saying that 5 times fast. And if those were made feasible, they could be used to get fluorosensors out in biplanes. Now, make no mistake, they are expensive. They can be up to $500,000 each. But on the scale of government, of like a small fleet of this, there have been worse purchases, and this could possibly be used to actually help in some emergencies. So let's move on to other potential applications. So first of all, rapamycin. That's a immunosuppressant used in cancer treatments and such. And it's important to know if it's in somebody's body because it's an immunosuppressant, and you need to know if you need to get it out. You need to know a lot of stuff. It just, a lot of stuff depends on whether it's there or not. So how can we find out if it's there? Well, through a process called quenching, which I will describe very soon. It can also be used to find other chemicals in the body, some iron, some metals, just some other stuff that's in the body that can be used using fluorosensors in the technique I'm about to tell you about. Quenching. Quenching is any process that makes the intensity of fluorescent light go down. So there are a bunch of processes to make this happen, but the most important one for this is some interactions between molecules, specifically in this case, biological ones, where some electrons move around and it changes the wavelength of the emitted light, the fluorescent light. And this is going to change, and this is measurable. So this can be used to determine if the targeted molecule, rapamycin or some other biological molecule, is actually there. And that can be used, like I said earlier, to figure out if you need to remove the chemical, a bunch of stuff, but it helps you know if it's there. So that is it. Thank you for watching, and I don't know who's watching this specifically, professor, and I don't know if it's students, but either way, thank you for a wonderful year, and I hope everyone who watches this ends up in good spirits soon and has a wonderful day. So thank you. I hope you enjoyed this presentation as much as I did. Bye.